I was going to start with a quote from one of the most famous statisticians, which goes something like, if you're going to go to a statistician after you have conducted your experiment, the only thing that the statistician will be able to do is tell you what the experiment died of. He won't be able to help you. So the take-home message of this statement, which was made in the 1930s in the context of crop science, is that, well, hopefully statisticians can be helpful in the process of evaluating interventions, and talking to them sooner than later is a good thing. The other reason why I particularly like this quote is because it very well describes what I do. I think about how to collect data. I think about how to analyze the collected data, but I very rarely actually analyze the data once they've been collected. So my entire research focus really is on thinking about how do you design clinical trials or studies more generally. Now, at the same time, I'm also very much aware that statisticians sometimes are probably the easiest to work with. I'm sure you've been on meetings with statisticians in the past where the only response that you've gotten was, "Oh, can't do that. Ooh, that's tricky. Oh, that's going to be a problem." And obviously, especially in the context of an outbreak, in the context of a pandemic, this is potentially very, very problematic. So here's my quote in full that at least hopefully semi-cited it correctly. Can you go to the next slide?
Yeah, I'm just going to see if I can put it in the full screen mode. Full screen mode. There you go. Okay, is that better?
Looks good for me.
Fantastic.
So here is a summary of trials during the 2016 Ebola outbreak. So what you can see on the x-axis in this graphic is time. On the y-axis, you see the number of cases that have been reported, and you can see two different colored lines in this graph. These are just two different reporting mechanisms of the number of cases that have been used here. So you can see somewhere around November, December time, there's the peak in the outbreak, and then the epidemic, fortunately in this case, seems to have ebbed off relatively rapidly thereafter. Now, in addition to the number of cases, you can see a couple of places are highlighted with the arrow, and these arrows correspond to the time points when different clinical trials started during that outbreak. So the first bigger trial that was started just about at the peak of the outbreak. So convalescent whole blood was started at around that time, and pretty much all the trials thereafter are already on the downwards trajectory of the outbreak. And so my very first learning or the very first reaction when you see something like this is, well, pretty much all of the trials have completely missed the boat, right? Because only one of the trials, you could argue, started when cases were still increasing. Everything else started after the peak of the pandemic, and therefore had very little or no chance whatsoever to answer the question that they were intended to answer. Next slide, please. And so one of the first lessons, and one of the first things to think about for a trial during a pandemic, it must start quickly. And that's not only so that we'll be able to find an answer to the question that we are posing, but also there's a potential to help the patients quickly, of course, but also if we manage to find a treatment that's effective quickly, we can actually use that treatment as part of the outbreak response and therefore influence how the further trajectory of the outbreak is going to look like. At the same time, obviously, there are lots and lots of uncertainties, especially in a context where there's a new disease, an unknown disease. We have no idea which treatments are potentially of interest. We don't really know, or we didn't know for the COVID pandemic, what kind of outcomes would be relevant, and so on and so forth. And then thirdly, during an outbreak, during a pandemic, the frontline workers are under immense pressure. They probably don't want to spend an extra hour filling out forms and data collection sheets and whatever else in order for you to run your clinical trial. Next slide. So with this thinking at the back of our head, when the first cases of COVID appeared, we started thinking about, okay, how could we avoid some of these pitfalls? And what came out of that is what's known as the Lotus trial. So the trial started on the 18th of January 2020. So that's about 19 days after the first official report of COVID. Now, therefore, because we had set it up to start as quickly as possible, we had to make some rather hard decisions and potentially not optimal decisions. So, for example, we couldn't just wait for lots and lots of evidence about potential treatment options. We had to go with something that had some potential, but obviously the knowledge base at that point was rather limited. So we ended up going with an HIV treatment, lopinavir-ritonavir, because it showed in vitro, at least, that it had some potential. We didn't know exactly what the right or what a good endpoint for the trial would look like, but we had some information from other studies in influenza on which we built. And so we started planning the trial with rate of clinical improvement as the primary endpoint. But probably most importantly, we didn't spend the very limited time to get the trial started on thinking about, okay, how many patients are we going to have to recruit now? But instead, we focused on getting a process in place that allows us to collect information, meaningful information, randomized information quickly. And once that process is in place, we started filling in the rest of the study. Next slide. And so subsequently, the trial was adapted. There was a change in the primary endpoint. There was a bit more understanding, appearing a bit more discussions with other stakeholders of what a meaningful endpoint would be. And so there was a change prior to looking at any data to time to clinical improvement rather than the rate that I'd shown on the previous slide. We've also supplemented the trial with a formal sample size calculation and so on and so forth. But the important part for us was get the trial started, get meaningful data collection processes in place, and then fill in all the blanks afterwards that you don't need at the onset of the trial. Now, as many of you probably know, the trial was run in Wuhan, and the control measures meant that the trial never recruited to the full target, but it gave us a lot of learning to think about, and especially once it became apparent that this is not going to just be a local problem, but is going to become an international issue, to think about what would the perfect trial during a pandemic look like. Now, this is my personal list. I'm sure there's lots and lots of other things that everybody here can add to this list. But for me, key aspects include using a meaningful and efficient endpoint. It should be able to make decisions quickly about treatments that aren't working particularly well. It should be able to cope with multiple treatments, especially in the light of we're starting off using repurposed treatments. We don't have strong evidence that either of these will work particularly well in this patient population, this disease, and therefore we want to give many different options the opportunity. We want to minimize the burden on frontline staff, and so on and so on. Next slide. But probably the most important aspect is that because of the amount of uncertainty that we have, the trial design needs to be adaptive. And so it needs to have the opportunity to make changes to your plan, obviously informed changes to the plan as the trial goes on, rather than sticking with the traditional approach of, okay, we've done the design, we've set everything, written everything in the protocol, and now we're going to wait. We're going to sit and hope that we've got everything right. Next slide. So out of that thinking came what's become the recovery trial, which is a multi-arm platform trial. The endpoint was chosen on very pragmatic grounds. I don't think anybody would argue that mortality is a meaningful endpoint. And it used minimal data collection, extra data collection, and made a lot of use of data linkage. Next slide. And so the trial, just as a snapshot, how it progressed over time, it started off with 4 different treatments versus standard of care. First adaptation was about a month into the trial. An additional treatment, tocilizumab, was added to the trial. Next slide. A couple of weeks later, another treatment, convalescent plasma, was added. Next slide. Then first decisions were made. Hydroxychloroquine was shown to be not promising, not sufficiently promising to warrant any further investigation, and so on and so forth. And so you can see that with this structure, with this platform structure of the study, we are able to investigate multiple questions at the same time. We started off with 4. By now, I think we're up to 14 questions that we are investigated or have investigated within the study. And it is set up in a way that allows decision-making on each of the questions that you have as information accumulates. Next slide. And I think it's fair to say that it's been a great story. It's been a great story in terms of recruitment. Within the first 90 days, almost 12,000 patients have been recruited. A lot of different hospitals and centers contributed. Trials open to all comers, all ages. It was very quick to go from funding decision to running the trial, and so on and so forth. But I don't want to just tell you how fantastic of a job the recovery team has made, because obviously there's always things that one can do better. Next slide. And so one of the questions that has been bugging me, or that that I have spent the last couple of 18 months or so thinking about is, so how should you add new treatments into the study? When should you include them? And what do you include in your study? So the how is more of a technical aspect. We want to make sure that our decisions that we are going to make about each of our questions is still valid, isn't biased in any way, and so forth. And so we're thinking about the statistical aspects of how should we do this. The when is both a statistical question, but also a very pragmatic question. Of course, the easy answer would be, well, you include promising treatment as soon as you can. But obviously, there is a risk that by adding additional treatments to additional questions to your trial, you may end up answering no questions at all. In recovery, what we've done to avoid this problem was to include new treatments in a factorial manner, so that patients would receive multiple active treat or had the potential of being randomized to multiple active treatments, but that causes or could result in additional questions around, okay, can you really distinguish the effect of the one from the other treatment? Another aspect that was very unusual for the trial and definitely raised some eyebrows was that there was no formal stopping rules within the study. So when the trial was started, basically the goal was recruit as many patients as you can and let the data monitoring committee make decisions about when to stop a treatment, either because it's looking very good or because it is not performing well enough. This puts a lot of responsibility on the DMC, who are to some extent trying to step into the role of the investigators and make decisions for the investigators. And so it's probably something that in this specific instance was justified to do, but probably isn't an approach that one could use, generally speaking. Another aspect of the study was that because mortality was used as the endpoint, it required very large sample sizes in order to answer the questions that were posed. Now, it did answer these questions very definitively. There was no argument that once you had an answer, that this was almost certainly the correct answer, but it still required thousands of patients to be recruited. And obviously, the list of things goes on. So on the basis of that experience and the thinking behind some of these issues that I've mentioned before, when we started thinking about the HEAL COVID trial, we tried to take some of the learnings from the recovery trial and hopefully come up with a trial design that's even better than what has been used or is being used in the recovery study. So, for example, rather than using mortality yes or no binary outcome, we use a time to event endpoint, which, because it contains more information, gives us requires smaller sample sizes in order to reach a conclusion. The trial includes formal sample size calculations to some extent based on better understanding of the disease, better data around the disease, but also to make sure that it is clear when and how decisions are going to be made. We also included a strong guidance for stopping if a treatment isn't performing well enough. And the thinking here is, again, the Data Safety Monitoring Committee is stepping in for the investigators to make some very, very crucial decisions. And having sat on these committees myself, it is very, very difficult to make a decision about giving up on an intervention, especially if you are going to make the decision for somebody else or for a different group. And so we felt that it was very important to give some very strong guidance, at least when we would stop the study. And finally, we've decided that we didn't want to run an excessive risk of not answering questions that we were posing, and therefore restricted the number of comparisons, the number of questions that were looked at the time to a maximum of 3. So there's still the potential to add new treatments to the trial, but if there are 3 investigations already ongoing, then this new treatment will have to wait until a decision is made on one of the other treatments. Next slide. And some of that thinking, some of that reasoning why we constrained the number of questions to be looked at has to do with the Ebola experience, where, as I said, fortunately for the people, unfortunately for the questions asked in the trial, the epidemic or the outbreak stopped before most of the trials could answer the questions they were set out to do. Now, with COVID, we will have, or we are having the same problem. When I last checked in November last year, there were over 2,200 trials running worldwide and just under 100 trials running within the UK investigating treatments for COVID in various different populations and stages. As you can imagine, with that, there's a huge risk that once there are no longer sufficiently many patients to be recruited, you will end up with lots of trials that will not be able to adequately answer the questions that they were seeking to answer. So you have a risk that you make decisions on underpowered study. You have a risk that as a consequence of that, potentially useful treatments might be disregarded and be thrown out. And there's been a relatively recent proposal, next slide, how one could think about countering that, that risk. And it's a collaborative alternative, which works effectively in the following way. You have one core protocol that's similar, ideally the same, across a lot of different potential sites. If you happen to end up in the situation that your location, your study runs out of potential patients, you use adaptive methods, specifically alpha spending methods, to check, do I have enough evidence already to make a definitive conclusion? And if yes, that's great. But if not, then rather than releasing the results of the study to the public and potentially harming a potentially useful treatment, you keep the results blinded if there is no definitive answer possible at this stage. And you would restart that protocol at a next outbreak or in a different location if that's more suitable. With that, I'm already at my end. Just to highlight, I do genuinely think that randomized controlled trials are the gold standard for evaluating treatments for evidence gathering, and the same applies within a pandemic. Next. But if we're working in trials for a new disease, then these studies should be adaptive because of the amount of uncertainty that we have when we are planning these studies. We cannot just plan a study, sit back and relax and hope that we got everything right. We need to be able to make the changes. In the context of a new disease, mortality clearly is a desirable primary endpoint, but because of the numbers required, it might be difficult to use in practice. However, time to improvement, time to recovery type endpoints have been shown to be quite useful alternatives instead that require much larger, much smaller, I should say, sample sizes. And finally, for every trial, but in particular during outbreaks and the pandemic, collaboration is crucial. Thank you.